Hello guys, welcome back to our channel Biolab, and this is Vamsi. Yeah, hope you all might wonder that I would start the MCQ series. I said at 6 o'clock, but you can wonder that why I'm here. Yes guys, before starting the MCQ series, I have got an idea that okay, let's start at 6 we will be having MCQ series, but in the morning session we will be going through the NCERT guys, so that it would be, it can be revisable to you, it would be revised. Okay, the chapter which we are going to conduct the MCQ series would be revised. Yes, the 30 under 30 MCQ series would be starting from today itself, and that is 6 at 6 p.m. Yeah, hope you all join, and this session might help you in revising the concepts. Okay, this is your NCERT series. This 30 under 30 means 30 MCQs under 30 minutes guys. Okay, 30 MCQs under 30 minutes will be completing the MCQs. Yes, this session every day we will be, I hope we have completed 3 chapters. Yes, so that is why we will be having this same pattern for 3 days. Morning sessions we will be having the NCERT series, and then in the evening session at 6 p.m. sharp you will be getting the MCQ series video. 3 to 4 days we will be taking, 3 to 4 days we will be taking to complete the NCERT series and MCQ series. Yes, if I have no time with me in order to do this NCERT series, then you can go on reading yourself guys. If I'll be telling you when I'm not free, then I'll not be uploading the video, but if I am free, I'll be doing the NCERT series guys. Yes, let's start. Today we will be discussing the chapter Microbes in Human Welfare, as we have the MCQ series on that today. And this in this chapter, as per your NCERT, these are divided into 6 subtopics. 1. Microbes in household products. 2. Microbes in industrial products. 3. Sewage treatment. 4. Biogas production. 5. Biocontrol agents. And 6. Biofertilizers. Yes guys, this series would be a fast phase series because we have already completed this chapter. So I'll be just going on reading the NCERT and underlining the important points so that you can also underline. So pause the video, get your NCERT, then set in front of this video guys. Then it will be much clear to you all the doubts. Yes guys, if you want the detailed videos on this, we have our playlist. Go there in our channel. I'll be pasting the link in the description box also, or in the iCards there, so that you can go and watch the playlist. Yes guys, let's start. Yeah, I have underlined some points for you. Here the point says microbes are diverse. There are different forms of microbes in the in the universe. One form is as they are present as protozoa, bacteria, fungi, microscopic plant viruses, viroids, and also prions. This is very important guys, please underline this point. It is in your first page of this chapter. You go there and underline this point. Prions are that proteinaceous infective agents. Prions that are proteinaceous infective agents. Let's write. Prions contain only proteins guys, as their the RNA and DNA are not present, but only proteins are present. Yes, we have seen the cell proteins. What are they? Histones. Yeah, histones. Like that, they only have proteins. Okay, proteins in them. Prions, proteins. Remember, like this P square. This question might appear in your MCQ series guys. Yeah, I'm revealing the questions here. Let us not reveal the questions. Let's move on. Microbes like bacteria and fungi are grown in nutritive media to form colonies that are being seen with our naked eyes. So these microbes and bacteria, what they do is, if you put them in nutritive media, they grow into colonies. They become a bunch like this. Yeah, have you ever seen algae growing in the stagnant water? See how that algae grows. We can see that algae with our eyes, literally like this. This is the culture media. The culture medium is a nutritive medium, and if you study this cultural medium, it is enough to study about microbes which are growing in that media. Okay, they are grown in nutritive media. So now that is about the first page. I haven't put much thing about in the first page. Yes, 1 minute guys, page is loading. This takes time. Yeah, these diagrams are very important guys. They can ask you in your examinations. If you are not able to see, let me zoom out a little. Yes, these diagrams are very important. We have to learn them. See, we have discussed various shapes of bacteria in living world. I hope I haven't discussed here. In that channel I have discussed on living world. Then we have discussed various types of shapes of bacteria: rod shaped, spherical shaped, spiral shaped. So those all we have discussed there. Now let's see this. These are rod shaped bacteria. See a picture. Guys, please, if you have your NCERT, write it side rod shaped, or you can put rod. These are rod shaped. Next one is spherical shaped. See, they're in balls, like they're like this, see. So these are called as spherical shaped. SP, you write SP means spherical shaped, you write the full file. I don't have space here, so I am not writing. Please do not go with the matter sorry, which is here. You can miss that one. So that is why I am asking you to write aside. See, rod shaped, spherical shaped. This is again rod shaped bacterium which is with the flagellum. If you find it, see, it is like rod, right? If the bacteria shape is like rod, that is rod shaped bacteria. If the bacteria shape is like spear, they are spherical shaped bacteria. So then we will be discussing there in detail. Now coming here, this is very important diagram of a bacteriophage. It is a very important diagram of bacteriophage. See, this part is your head part. This part is your head part. This part is your the diamond shaped thing is your head part. The below that one is collar part. Same like here, collar. Below the collar part, this one is your tail part, and this is whole structure is present on a plate like thing called as a plate, and this plate gives rise to legs called as prongs, and it also have pins in order to attach onto the substratum or the surface. So these are your pins. See here, these are your pins. Okay, those are your pins. This is your head, collar, prongs, pins, tail. Okay, got it. It is a diagram of bacteriophage. Right, bacteriophage, right back. Now coming to the next diagram, this is adenovirus, guys. This can be asked in your NEET exam. Please write it. Adenovirus. Adenovirus. Okay, this is your adenovirus, guys. This B color B picture is, write it there. Now this C1 is tobacco mosaic virus. It is rod shaped TMV. Tobacco mosaic virus. It is called mosaic virus because the pattern of it is mosaic pattern. So we'll be discussing in some lesson. When we go there, I'll be talking about this tobacco mosaic virus. For now, remember this is rod shaped TMV. They can ask anything, any picture, and if they ask the bacteriophage, they can ask the labeled parts, or if they ask adenovirus, they can give you the picture and they can say identify the picture, or if they can give TMV and give you identify the picture. So they're like that they can ask anything. These are bacterial microbes, these are viruses. Okay, viruses. Now let's move on. These are your colonies. See this, these two are the colonies. This is your culture medium, which is nutritive medium, nutritive in nature. Nutritive in nature. Yes, this session is going to be a little lengthy since we have to complete 12 pages of your NCERT. This A color, A picture is your bacterial colony, and B picture is your fungal colony. Okay guys, yeah, let's move on to the next page. Now let's start your first part from your NCERT, that is microbes in household products. This is very important. From here please learn everything. Now coming here, these microbes are useful in our day-to-day life in many ways. These microbes are even present in our digestive tract. They are even present outside. They are even present in the atmosphere. They can cause diseases. They can be harmful microbes. They can be the microbes which are helpful. So we cannot think all microbes are harmful, but there are helpful microbes, such as lab bacteria is one of the bacteria which grows in milk, and it helps in the conversion of milk to curd, we have discussed. And during the growth, this lab bacteria produces acids, lactic acid, which coagulate and partially digest milk proteins. This is very important point. Now a small amount of curd is added to the fresh milk as an inoculum or a starter, which contains millions of lab bacteria. This is called as inoculant or inoculum. From this inoculant only the lab bacteria is gonna invade into the milk. Okay, this a small amount of curd is the previous day's curd which you have put to for the conversion. So that is what. Now these lab bacteria, they increase the nutritional quality of the curd by increasing the content of vitamin B12. So that is why curd is more nutritious than milk. Curd is more nutritious than milk. Got it? Now this is your first use. Of lab bacteria. Let me write it here. 1 minute. The first use of bacteria is lab. I am writing it here. Milk to curd. Hope you are sitting with your NCERT. Yeah. Before 1 minute guys, let me give you a small hint here before proceeding further. Yes guys, before watching the MCQ series, please do watch this video and then move on to the MCQ series. That may be helpful to you guys. One thing. And then if you are coming to the NCERT series, you have to bring your NCERT series, sit and underline the points either with the help of a highlighter or with the help of a pen or with the help of a pencil, and you have to write the points sideways. See, like this. There will be place in your NCERT. I don't have my NCERT with me present, so if I have, I would have shown you my NCERT how I am doing it, so that way we can get benefited guys. Yes. Now next use of microbes is in the preparation of dough. It is used in making breads. The dough is used in making chapatis. So that dough is fermented while preparing idli and dosa guys. Idli and dosa dough is fermented, but the dough which we use in chapatis, that is fluffed. Okay, that is fluffed. So the fermentation of dosa and idli dough, we cannot call it as dough, but we can call it as batter. Idli and dosa batter is done by Muconostoc mesenteroids. We have already discussed. It is not mentioned in your NCERT. They only said that fermented by bacteria, but just remember the name Leuconostoc mesenteroids. Okay. Now the puffed appearance of the dough is due to the production of CO2 gas. See, while making the pizza, in order to make the pizza base, we add activated yeast into the dough while mixing the dough. That brings the puffy nature to the dough, puffed appearance. This is due to the release of CO2 gas, and this CO2 gas would go and sit into the holes of this dough, and it makes the dough puffy. So that is why we use yeast, and this is fermented. This is the process is done by fluffing process is done by baker's yeast, baker's yeast. Okay, Saccharomyces cerevisiae. Why are we saying as baker's yeast and beaver's yeast? So in order to ferment a number of drinks, we have a number of drinks coming. Next, alcohols. In the preparation of ethanol, the same yeast is used. So that as a common name we call it as brewer's yeast, and this as a common name we call it as baker's yeast. Both are Saccharomyces cerevisiae only. A number of traditional drinks and foods are also fermented by microbes. This traditional drink example is toddy. It is fermented by some natural microbes, and I'll be telling you the name when we are doing the MCQ series. Yes, there we have a question which is not given in your NCERT, the name of the microbes. I don't not remember the name. Yes, of the microbes I'll be giving you there, and that I thought of saying no, but I do not remember the name. This toddy, it is a traditional drink which is fermented by some natural microbes, and it is collected from the sap of palm trees. Now, microbes are also used to ferment fish, soya bean, bamboo shoots to make foods. Now, cheese is one of the most oldest food item which is in which microbes are used. Different varieties of cheese are known by their characteristic texture, flavor, taste, specificity of the microbes used. Now here it is very important. Different varieties of cheese are known by texture, flavor, and the specificity coming from the microbes used. Very important. Very important, that part. Now, for example, large holes in Swiss cheese are due to the production of large amounts of CO2 by a bacterium named Propionibacterium shermani. This is very important name, Propionibacterium shermani. And Roquefort cheese is fermented or ripened using a fungi called as Roquefort fungi. Okay, Roquefort fungi. Now, this is the use of microbes in household products. Now we will be moving on to the use of microbes in industrial products. In industries, microbes are used in synthesizing many products like alcohols. Here only we will be discussing the use of microbes in medicinal industry only, and this first we will be talking the production of alcohol or ethanol on large scale, and it is done in a vessels called as fermenters. This production of ethanol is done in large vessels called as fermenters, and interestingly our friend Saccharomyces cerevisiae yeast, brewer's yeast, is used in the fermentation process. Now let's move on to the second page. See, on your left side page figure number 10.4 are your fermenters. These are your fermenters. See how big these vessels are. In this our alcohol is being fermented. These are your fermenters. Okay, now let's see. Microbes, especially yeast, have been used over a time for the production of beverages like wine, beer, whiskey, brandy, rum, gin, etc. For this purpose, the same yeast, remember here the same yeast, Saccharomyces cerevisiae, but we call it by a common name called as brewer's yeast. We call it by a common name called as brewer's yeast. One minute. We call it by a common name called as brewer's yeast. Now, used in the bread making and commonly called as brewer's yeast. In the bread making, that yeast is called as baker's yeast, and here it is called as brewer's yeast. Now, is used in the fermenting of malted cereals. Malted cereals. This is what to say. I don't know. I pronounce it as cereals. So then let us continue. Malted cereals and fruit juices to produce ethanol. To produce ethanol, the main base, the main base nutrient media for the microbes or the yeast in order to grow and ferment this ethanol or alcohol is your malted cereals only. Malted cereals like barley malt, barley cereals. So those are your nutritive media which help these yeast microbes to grow. Now, depending upon the type of raw material used, this is very important, depending upon the type of raw material used for the fermentation and the type of processing, here the type of processing means with or without distillation process. If with a distillation process, there are separate alcoholic drinks. If without distillation, there are separate alcoholic drinks. These are very important. Wine and beer are only the alcohols which are produced without distillation. Wine and beer are only and only the alcohols which are produced without distillation. But whereas whiskey, brandy, rum, gin, etc., these all are produced by distillation. But both are by fermentation process only. Both are by fermentation process only. And here you can see the fermentation plant. So this figure 10.5 shows you the fermentation plant, and figure 10.4 shows you the fermenters. Now let us discuss the use of microbes in medicinal industry. Okay, in medicinal industry. So this is all not necessary. The necessary thing here is anti is a Greek word which means against, and bio means life. Together it means against life. Antibiotics means against life in Greek. In context, that is in context. Against life means not against to our life. They provide security to us and they fight with the microbes, disease causing microbes, disease causing microorganisms. Whereas with reference to human beings, they are called as pro-life, not against. Okay, they are called as pro-life. No. Let's move here. See, the first antibiotic discovered was penicillin. We all know. It was discovered by Alexander Fleming. And here. Penicillin from Penicillium notatum. Penicillin was the first antibiotic discovered from Penicillium notatum. We remember this by Alexander Fleming, but we will not remember that this was an what accidental discovery, and it was done when Alexander Fleming was working on a bacteria called as Staphylococcus or Staphylococci. We will not remember this name. We will be remembering these three. These three we will be remembering, but we will not remember this name. So the question may appear here. The question might appear there. The first antibiotic penicillin discovered by Alexander Fleming was an accidental discovery while he was working on. They will give options: Penicillium notatum. We will be remembering Penicillium notatum. We will be remembering penicillin. Then we saw penicillin. Okay, Alexander Fleming. There is another word called as Penicillium notatum. Let's mark that. Completed. Answer gone. They will be giving you Staphylococci as your last option. Then what you will do? So you will lose the mark. So not like that. We have to remember this. Alexander Fleming, Staphylococci. When he was working on Staphylococci, he found penicillium using Penicillium notatum. When he was working there, he was culturing this Staphylococci bacteria in a nutritive plate, and on that plate there was some random spore which was you know in the atmosphere, landed on that plate and grew, multiplied its colony, and then when it multiplied there were no bacteria growing there, and it eradicated all the bacteria if it is put on the next plate. And that spore was interestingly of Penicillium notatum, and from that name he named it as penicillin. But even though he found this penicillin, there was no recognition for his work until Ernest Chain and Florey. You can remember Chain and Florey. Until Chain and Florey brought this antibiotic into use. This antibiotic was extensively given during the time of World War II to the scientists who got to the soldiers who got injured. And if they were given this drug, the next day morning they came again to fight. Their wounds were vanished. So that is why they called it as wonder drug during that time. So therefore Fleming, for discovering this penicillin, Chain and Florey, in order to bring this penicillin into the usage, were awarded Nobel Prize in the year 1945. So completed. Moving on to the next page. Now, after penicillin, there are other microbes, other antibiotics are also discovered, and some antibiotics like azithromycin, which is generally used for cough. Azithromycin, yeah, those are also discovered. Now, let's see chemicals, enzymes, and other bioactive molecules. So let's see the use of these microbes in preparation of chemicals, enzymes, and other biomolecules. Now, Aspergillus niger or niger, a fungus, it is used in the preparation of citric acid. Please write somewhere on a different paper and stick it onto your NCERT textbook. Write these all. So first put the heading. 1 minute, let me show you how to write this. See, put like this: Aspergillus niger. I am writing the short form. Put an arrow mark: citric acid. Aspergillus niger. Acetobacter aceti. It is a bacterium which is used in the preparation of acetic acid. Right here, Acetobacter aceti, used in the preparation of acetic acid. Okay, Clostridium butyricum. It is used in the preparation of butyric acid. It is a bacteria. Lactobacillus, used in the preparation of lactic acid. Now, these microbes not only useful in the medical industry or not only useful in the chemical industry, they are also used in making enzymes like bacterial lipases, which is used in the detergent formulations. Lipases are used in the detergent formulations, and they are helpful in removing the oil stains from clothes. If you give it to the laundry person, he will be putting a detergent which contains a bacterial lipase, and it helps in the removal of those oil stains. Okay. Now, bottled juices are also very clear. With they are cleared with the help of pectinases and proteases. Bacterial pectinases and bacterial proteases or enzymes which help in the clearing of those or filtering or those suspended particles in the bottled juices. Streptokinase. This streptokinase produced by a bacterium Streptococcus, and genetically modified engineering, it is used as a clot buster. Actually, what is this streptokinase doing is it it releases the clots. If there is any clot in your heart, heart is this I'm showing here. Yeah, if there is any, any blockage of blood in your heart artery, if you get a streptokinase injection, then it is going to bust that clot, and therefore it removes the clots. So that is why it is called as clot buster. Now another one, it is a cyclosporin A, and it is an immunosuppressant agent in organ transplant patients. Why, you can ask me, why they are using immunosuppressant cyclosporin A? They can use any other immunosuppressant, yes, like steroids. Yeah, these steroids are harmful to our health, but this cyclosporin, when it is given in a limited quantity, it is not harmful to our health, and it is obtained from the organic, you can say living organisms, from living organisms. So that is not much effective, like it is not much effective on our health. It doesn't show any side effects. So that is why a fungus Trichoderma polysporum produces is produced, sorry, what I am saying, this cyclosporin A is produced from Trichoderma polysporum. Statins, these are again, they are produced by yeast Monascus purpureasis, and they have been commercialized as blood cholesterol lowering agents. These statins work upon the principle called as competitive inhibition. Here we have, they work on the principle called competitive inhibition in order to reduce the cholesterol level in our heart. They are defending or preventing the synthesis of cholesterol. So competitive inhibition, stoppage of enzyme responsible for cholesterol synthesis. Now we have completed the microbes in household products. We have completed use of microbes in industrial products. We have completed microbes in the production of chemicals. We have completed the microbes in enzyme production and bioactive molecule production, and also we have completed in the medicinal industry. Now we will be seeing the use of microbes in sewage treatment plants. These sewage treatment plants, before going, let's see the meaning of sewage. This is the meaning of sewage. So this is the meaning of sewage. Okay, let me take marker. See, this is the meaning of sewage, and this sewage contains much organic matter and microbes. If you send this sewage without untreated sewage, or if you release into the rivers, this sewage can contain effective pathogens which cause diseases to the river bodies, and which are these river bodies become prone to diseases, and they can kill small fishes. So in order to stop this untreated sewage releasing into the waters and getting them contaminated, our government has prepared something with the use of microbes called as sewage treatment. We treat this sewage before the disposal into the water, and this treatment is done in sewage treatment plants called as STPs, and making the water less polluted. Okay, this treatment is done by the use of microbes. Let's move on. This is very important. Those microbes are heterotrophic. They can ask you, they can confuse you in your exam anyway, but you have to only choose this. So let me reveal a secret to you. Okay, let's keep that secret only. When you come to the MCQ series, then you will be understanding that. Yeah, heterotrophic microbes. The treatment is done using heterotrophic microbes, and these are naturally present in the sewage. This treatment is carried out in two stages. One is the primary treatment, second one is the secondary treatment. In primary treatment, physical removal of particles like large and small sand particles are removed from the sewage through the filtration and sedimentation. So filtration, you will be having filters on the, if this is the pipe, you will be having the filters, and when the sewage is rushing down, this these filters stop the large grain sand particles, large particles, and this cleared sewage is flown into a tank, and they are used to settle down, and this is called as sedimentation, moving of heavier particles down and lighter particles up. And after this sedimentation is done, the water which we get, the sewage or water which we get, is called as primary effluent. So this, yeah, the upper part, if this is your sedimentation tank, in this tank, if this is your sludge, this is your effluent. This water is then passed into the next tank which contains aeration, then we will be seeing it a little later below this. And this this black color one is called as primary sludge, and that is what is the physical waste material. Okay, physical waste. Now after settling, this primary effluent which is produced in the primary treatment is then flown into the secondary treatment tanks, in which large aeration tanks are present, and here this sewage is continuously aerated or agated mechanically by the passing of air into it. This is called oxidation tanks also. They are called as oxidation tanks also. Okay, this allows vigorous growth of useful aerobic microbes into the flux. Now what we do is, if you put this into the sedimentation, the oxidation tanks, we pump air into these oxidation tanks, and we add a little amount of aerobic microbes which contain bacteria and fungi, and with the help of these, as they are aerobic microbes, with the help of this air, they grow their colonies and form flocks, bacterial flocks. So like this, these are the bacterial flocks, and they decrease the BOD. They decrease the BOD. You have to remember aerobic microbes. Now, while growing, these microbes consume a major part of organic matter in the effluent and significantly reduce the BOD, biochemical oxygen demand. We have seen that BOD directly proportional to organic matter, directly proportional to pollution. I'm trying to write it without seeing onto the tab, so that is why it is coming like that. Yes, this you have to remember. BOD directly proportional to organic matter, directly proportional to pollution. Very important. Now, this sludge which is produced here, not the sludge, no, after this aeration, there will be bacterial flocks floating here and there in the effluent. So in order to make that bacterial flocks settle down, so we put it in the sedimentation tanks or settling tanks, and this bacterial flocks settle down and the water comes up, and now this water is treated well and it can be released into the rivers, and this is called a secondary effluent, and the secondary effluent is released into the rivers, and this flocks which are produced here is called as activated sludge, and a small part of it is added to the next procedure when the primary effluent is put into this. As I said, we will be adding the aerobic bacteria, aerobic microbes that comes from the previously activated sludge, and some part is used for that, and the remaining part, it is pushed into the anaerobic sludge digesters. As their aerobic sludge, as it is aerobic sludge, in order to digest that aerobic sludge, we use anaerobic sludge, anaerobic bacteria. this activated sludge contains aerobic bacteria. We have seen here, see, aerobic bacteria. I have told you to remember. Now in order to digest that one, in order to finish that one, we use anaerobic bacteria. Here we use different kinds of bacteria present, which are available in different regions, and this bacteria, while digesting this, they produce a gas called as biogas, and this can be used as source of energy, and it is flammable. It is flammable. If the bacteria is somewhere growing, like in the bread making industry, if you are producing biogas in the bread making industry, the bacteria uses yeast and it releases carbon dioxide. But if you produce the biogas from the gobar, it is also called as cow dung, and if you produce from that, it releases methane, methanogens. So like that, it is this biogas, it is a mixture of methane, hydrogen sulfide, carbon dioxide. But in the next page we will be seeing only hydrogen, not sulfide. They can ask you both hydrogen sulfide or hydrogen, you have to mark them, because the line is present has hydrogen sulfide. In the next page we will only seeing the hydrogen. Okay, got it? Clear? Yes sir, yes sir. Now let's move on. So about the microbes in biogas production, we have this one 10.4. Now let's move on here. In order to create awareness among the people, our governments like the central government have started two plans, action plans called as Ganga action plant and Amino action plant. Why did they decide to clear these two rivers? One, they are the major rivers of India. Two, we we see them as holy rivers. Ganga river we see at holy rivers, and STPs are being built on the banks of this Ganga river, like this. If this is your Ganga and Yamuna river, here are STPs, and these STPs clear. This is an aerial view of your sewage treatment plant here. That is your aerial view. I have sorry forgot to tell you this. Yeah, this is your secondary treatment plant, where your secondary treatment procedure is done. Remember this. So this is your aerial view of sewage treatment plant. Now let's move on to the use of microbes in biogas production. Use of microbes in biogas production. As I said, the availability of bacteria decides the type of gas released as biogas. If you have the bacteria in here, the bread making industry, the bacteria release CO2. Microbes release CO2. Like here, the same thing is given. The dough making industry and cheese making and the production of beverages are mainly produced CO2. That is why that contain what that contain. See, sorry for saying this, I am not feeling well. I have cold, I cannot talk much, so my energy levels are going down, so that is why I am running. So please do not worry. Yes, cheese, dough making industry, bread making industry, they use yeast as microbe in order to make the bread, so it releases carbon dioxide. So that is why if you produce biogas there, they use yeast only. So the same thing happens, CO2 comes as a biogas. Now if you use the bacteria which is anaerobically growing on the cellulosic material, produce large amounts of methane along with carbon dioxide and hydrogen. See, I said, in the before page they have written hydrogen sulfide, H2S. In this page they have written hydrogen, carbon dioxide, methane, CH4, CO2, H2 or H2S. Both are correct. Okay, or H2S. These are called as methanogens as they release methane, and common name is commonly found bacterium is methanobacterium. We remember methogens, we forgot methanobacterium. Then there will be there'll be a question in our NEET on methanobacterium. Then we think, oh, where I haven't read this methanobacterium anywhere. So that is why we have to. Now this is the common type of bacteria. Methanogens are used in the are used in the biogas production. What they do is when this methanobacterium is added to the anaerobic sludge digester, it digests, or it digests that aerobic bacteria and release a methane gas, and this methane gas, with the help of carbon dioxide and hydrogen, it is sent into the gas collecting chamber, and from there it is sent through a pipe into kitchens in order to use as a fuel. So this is also called as gobar gas, since it is produced from gobar. Gobar means cow dung. Okay, now this is your typical biogas plant. This is your sludge digester. This is a mixture of cow dung plus water. It is released into the digester. Here is the presence of your what you call, let me tell you, it is the presence of your aerobic bacteria or activated sludge. From here it flows here. Since it is also containing water, you can think like that, hydrogen is released. So here it is digested. The spent sludge is produced into here, the slurry, and this is your gas holder, and the gas is released here, methane, carbon dioxide, hydrogen or H2S, hydrogen sulfide. From here it is moved here, and from here it is produced into the rivers. Now the homes. Now this sludge which is produced here, it is used as fertilizer, and this gas is used for lightening and as a fuel. And this is developed in India by Indian Agricultural Research Institution, IARI, and Khadi and Village Industries Commission, KVIC. IARI and KVIC have in combined have done this experiment. You can say. Now, microbes in biocontrol agents. I hope we are close to completion. We are close to completion. We have last, if you complete this page, we have remaining one page. That's it. We are close to completion, guys. Let's complete it in 1 hour. Come on. Biocontrol refers to biological methods of controlling plant diseases and pests. See here, you have to understand the difference between biocontrol and biofertilizers. Biocontrol, they only control the diseases, means without spreading. Okay, in the modern society, these problems have been tackled increasingly with the use of chemicals. We are using chemicals, and this farming, which is done with the help of chemicals, is called as inorganic agriculture, or you can say chemical farming. By the use of insecticides and pesticides. These chemicals are toxic, extremely harmful to human beings and animals alike. Okay, now let's move on further. We use insecticides or pesticides in order to remove weeds itself. Weedicides. Now here, let us remember thing. What is this? A big written as 2,4-D. Let's write with pen. With pen also same thing. I'll be writing 2,4-D. At 2,4-D क्या है? Let's see. 2,4-D. Yeah, guys, we have discussed it is an auxin, synthetic auxin. Yeah, that is also used as an insecticide. And biocontrol and diseases. Let's see these microbes which are used in the control of pests. Now this is called as organic farming. Let's move on here. I am searching for the important line. Yeah, here is that. Very familiar beetle with red and black markings on the back called as a ladybird and dragonflies are used in the getting rid of aphids and mosquitoes. Ladybird, ladybird, aphids. LBA. Remember NBA? National Basketball Association. I hope I don't know the exact. LBA. Ladybird aphids. Ladybird aphids. Ladybird aphids. DM. Like CM, DM. CM, DM. Dragonflies, mosquitoes. Ladybird aphids. Completed. Ladybird is used in the control of aphids. Dragonflies are used in the control of mosquitoes. Okay. Now these another major problem is butterfly caterpillars. What these butterfly caterpillars do is they feed upon the plant leaves. They eat up the leaves. Have you ever seen leaf, original leaf or healthy leaf is like this, but the leaf which is eaten up by the butterfly caterpillars is somewhat like this. Originally its shape is like this, but it is being eaten up. So in order to control this, we use Bacillus thuringiensis, which is used in the control of butterfly caterpillars. In the control of butterfly caterpillars, we using genetic engineering, we have introduced this BT or Bacillus thuringiensis into the plants like cotton, brinjal, tomato, and they become resistant to the butterfly caterpillars. So they are called as BT cotton, BT brinjal, BT tomato, like that. Okay, in some parts of a country, in some states, it is even being cultured. Now, bio treatment in this biological control is developed for the use in treatment of fungus. This Trichoderma species, a free living fungus, it is very commonly used in rooted ecosystems, and there it is used to control many plant pathogens, several plant pathogens. Now, baculoviruses are the pathogens which are, an example of baculovirus is NPV. Remember this, it is enough. NPV, they are narrow spectrum. Now, NPV, how to remember this? NPV, nucleopolyhedroviruses, let's make it, it is generally NPV, let's add S also, NPVs, nucleopolyhedroviruses, they are narrow spectrum, see here, where is this? Yeah, narrow spectrum. S, species specific. N means narrow spectrum. S means species specific. Okay, insecticidal applications. Narrow spectrum, species specific insecticidal applications. NPVs. Narrow spectrum means narrow spectrum, species specific insecticidal applications. They only harm the let me tell you, they only harm the microbes which cause harm to the plant. They only kill the microbes, and they even do not go near the non-target insects, or they will not harm any mammals, birds, fishes, or even non-target insects also. So that is why it is specially desirable in the integrated pest management program which is conducted by our central government, and it is used there. Integrated pest management program, IPM. Now we have completed microbes as biocontrol agents. Now let's see microbes as biofertilizers. Yes guys, we are running out of time. I thought to complete this in 1 hour, so that is why I am running. Yes, now we have completed 1 minute. Yeah, see, chemical fertilizers and biofertilizers. The farming which is done with the help of biofertilizers is called as organic farming. It's called as organic farming, and this is very important. And we as a science student, I'm even getting you know yarnings also. And I have never taken this large session, 1 hour session, continuously speaking. So that is why organic farming, biofertilizers. Remember, it is very important. Now, the main sources of biofertilizers are bacteria, fungi, and cyanobacteria. This is cyanobacteria. They fix atmospheric nitrogen into the forms of into organic forms, and which helps in plant nutrient and plant growth. Plant nutrient and plant growth. Most of these cyanobacterial members can also be in legume plants. Sorry, what I am talking. These nitrogen fixers are also present in leguminous plants, and these leguminous plants which have association with the nitrogen fixer, and that particular nitrogen fixer, what I am saying, no, I lost my mind. The specific nitrogen fixer which is able to fix nitrogen in the leguminous plant root nodules is called as rhizobium. You have to remember this. Legume plants, rhizobium, LPR, PVR, LPR. Come on, let's do. Now, other examples of free living nitrogen fixers are azospirillum and acetobacter. Acetobacter aceti, used in the preparation of acetic acid. Acetobacter used used as a free living nitrogen fixer. Remember it. Yeah, guys. Sorry. Fungi. Fungi which are also known to form the symbiotic association with plants are called as mycorrhiza. Fungi symbiotic association. Okay, let's write like this: FMS, FMS, FMS. Fungi mycorrhiza symbiotic association with plant roots, and this genes is called as glomus, and they help in preventing root-borne diseases. So that is why they give the plants, they provide the plants resistance to root-borne pathogens, tolerance to salinity and drought, and overall increase the plant growth and development. They also increase the phosphorus absorption rate of the plant. Now, cyanobacteria are the autotrophic microbes widely distributed in aquatic and terrestrial environment, and many can fix atmospheric nitrogen. Example: Anabina, nostoc, oscillatoria, etc. In paddy fields, cyanobacteria serves as a very important biofertilizers. Blue green algae, it is also known as blue green algae. And why only in paddy fields it is used extensively? See, for the growth of cyanobacteria algae, blue green algae, we require stagnant water. So that is why in paddy fields we have stagnant water in order for the growth of rice plant. So that is why these can be used there extensively as a biofertilizer. So that is it, guys. Successfully we have completed our NCERT, and in the evening we will be discussing our MCQ series. Evening at 6. Please come join the session there. And this is Vamsi signing off. Have a great day further, and please do not forget subscribing to the channel and also liking the video if you like it. And also please do not forget, please come in the evening. Please, guys, please come in the evening. It will not be a live session, but you can watch the video. Watch this video first before coming there, since we have revised everything. So 3 to 4 days. This is a procedure which we will be doing. Next we will be starting a new lecture on genetics, guys. It takes much time, so let's complete it first. Genetics, will be starting principles of inheritance, then we'll be moving on to molecular basis. We'll be completing genetics, guys. First of all, it takes a large time if you put it to the last. So this is Vamsi signing off. Have a great day further. Thank you, guys. If you like it, please hit that like button and also subscribe to our channel. Please share the video.